Hi class, now we are on Unit 4 and it's all about nucleic acids. In this part, we will distinguish between DNA and RNA in terms of structure and function. In our next video, we will talk about the biological functions of nucleic acids in relation to replication, transcription, and translation, or simply the flow of genetics. Nucleic acids specifically are DNA and RNA.
If you've seen CSI or Korean novellas where the prosecutors collect hair samples, blood, or saliva in the crime scene and give this to forensic investigators, they will analyze these biological samples to get a DNA profile of individuals that the samples came from. DNA is deoxyribonucleic acid, and its major function is to contain the genetic information which stores the code for your proteins and exhibits your traits. Therefore, DNA is also called the blueprint of life or the recipe of life. It is very important, so it is stored in the nucleus and it never leaves the nucleus. DNA can also be found in mitochondria and chloroplast of cells. So if we look at the structure of DNA, it's a double helix, and its sugar is deoxyribose. The specific base pairing for DNA is adenine, thymine, and guanine, and cytosine. As a review, our nucleic acids are formed by millions of nucleotides joined together by phosphodiester bonds. So this is through the condensation reaction. This one strand of nucleic acid here is called the primary structure. DNA consists of a double strand of nucleotides. So the paired strands are coiled into a spiral called our double helix, and this is a secondary structure. And the structure can be inferred from Rosalind Franklin's famous X-ray crystallography. The sugar phosphate chains are on the outside, and inside you can find the nitrogenous bases, which are held by hydrogen bonds. So the two chains are held together by complementary base pairing. So if this is adenine, you have a thymine here. And if this is guanosine, you have a cytosine here. So between these, those that hold these together are your hydrogen bonds. And this base pairing can be inferred from Erwin Chargaff's research. Now we go to RNA. RNA is not that famous compared to your DNA, but RNA plays a very important role with connection to your genetic information as well. RNA is an abbreviation of ribonucleic acid. It contains the genetic information and transfers this genetic information from one generation to the next and functions for the synthesis of proteins. RNA is a single-stranded structure which can be found in both your nucleus and cytoplasm. Compared to DNA, the sugar of your RNA is ribose. And the nitrogenous bases include adenine, uracil, cytosine, and guanine. So it differs to DNA with this uracil nitrogenous base instead of the thymine. So RNA can be of three types. It can be a messenger RNA or mRNA, a ribosomal RNA or rRNA, and the transfer RNA or tRNA. The functions of these types of RNA can be understood more deeply when we go to the next video on flow of genetic information. But for now, what I want you to see is that the messenger RNA carries the instruction from our DNA to the ribosome, which we will see in um, transcription. And for rRNA, this combines with proteins to make up ribosome. So we will see this in translation. And tRNA brings amino acid to ribosome, which will also be in translation. So in this lesson, we distinguish the difference between DNA and RNA in terms of structure and function. So in terms of structure, our DNA is a double helix, and RNA is linear. In terms of the number of strands, DNA has 2 strands and RNA has 1. As you can see here, we have 1 strand, 2 strands. And for RNA, we have only 1 strand. In terms of sugar, we have deoxyribose for DNA and ribose for RNA. So you can see here the difference in sugar. Deoxyribose at C2' lacks that oxygen, and for ribose, it has that oxygen atom. In terms of the nitrogenous bases, for DNA we have adenine, thymine, guanine, and cytosine. For RNA, we have adenine, uracil, guanine, and cytosine. So you will see here the difference between the pyrimidines, thymine, and uracil. As for the function, DNA stores genetic information, and RNA here carries the genetic information and codes for protein synthesis.
Part of your textbook need not to be submitted, but this is important for you to know in the future to look at the differences between your DNA and RNA. So we've talked about that in the previous video about some of the obvious differences between DNA and RNA, but this table is a little more complicated. It shows you the differences in terms of the different types of RNA and their cellular roles. For activity 4.2.1, show and explain how exactly the condensation reaction of a nitrogen base and sugar results to the figure below. So you have to do the same thing what we've done in 4.1 examples, how the condensation reaction happened. So you may need additional sheet of paper but I need you to be very brief in your explanation. For activity 4.2.2, show and explain how exactly the condensation reaction to form a nucleotide happens. So it's similar to your 4.2.1, another example to get the hang of it on how this nucleoside and nucleotide are formed. For activity 4.2.3, you write the structural formula of GAC. We have done this in the previous PowerPoint video, write the complementary strand adjacent to it so that the complementary bases are side to side. So connect the appropriate base pairs. So we have the GAC. What is the complementary strand with the GAC? We've already done the first part, the GAC itself. Now you tell me what is the complementary strand. Remember that your guanine is only paired 2 for DNA. Your adenine is only paired 2 and near cytosine. So you do the that part, the complementary strand. For letter B, sticking to the convention of writing your nucleotide sequence in the 5' to 3' direction, what is the nucleotide sequence of the DNA strand complementary to ATG, CAC, CAT, GCT? So expect a lot of this in your quizzes and especially in your exam. So what have we learned from this unit? We learned about the differences between DNA and RNA in terms of structure and function, and we also learned about the different types of RNA.